Today I'm going to be talking to everyone about the development of a qPCR assay for hot powdery mildew, specific differentiation, and then how we can use that to understand late season dispersal patterns. So I will go ahead and give you a little bit of a brief introduction here. So hops, or Humulus lupulus, are a herbaceous perennial plant. They grow as a vine on an 18-foot trellis. And in late June and early July, they begin to flower. And you can see those cones up here in the picture at the top of the canopy. Those are used for their alpha acids, flavor, and aroma when brewing beer. So the pathogen that I'm deciding to hone in on is Powdery macularis, and that is the causal agent of hot powdery mildew. This fungus was first detected in 1996 in the Pacific Northwest, and it overwinters as an obligate pathogen, specifically on living host tissue. So mostly that's on crown buds, but it also will overwinter in the greenhouses, and when planting material gets shipped out in spring, that can cause infections in the field. So I'll tell you a little bit about the symptoms and signs here. So the fungus grows as a, grows as a white talcum powder-like substance. Fungus over here on the right in this cone. And when you zoom in on that on a microscope, you see these chains of conidia. And you're not always going to see the fungus on the cones, and what you're more commonly going to see is the browning here on the cone that reduces the color. Obviously, it should be a more vibrant green, and then also the alpha acid content that's used for bittering. So you will see these colonies throughout the year before flowering starts, but then as the leaves start to harden off, the infections will transfer over to the cones as they are now becoming susceptible and blooming later in the season. And in severe cases, you can have entire crop loss and this extreme browning that you see here that is very detrimental to the growers and costs them a lot of money. So to talk to you a little bit about the life cycle here, as it overwinters on living host tissue, it can overwinter as a chasmothecia on leaf litter and cone litter that falls down. And then it can also overwinter within the buds, both as mycelia and as chasmothecia. These chasmothecia will start to swell after a rain event in the spring and discharge ascospores, which will infect the younger plant tissue. Or if it's living as mycelia in the crown buds, when the plant growth starts to emerge, that mycelia will grow along with the flag shoot, or with the young shoot, and causes what we call a flag shoot. Both of these modes will then produce conidia. These conidia can reproduce and go into multiple generations throughout the year. Now, luckily in the Pacific Northwest, we don't have the overwintering chasmothecia, and the fungus is forced to overwinter in the buds. So Powdery macularis is heterothallic, but it only has the MAT11 mating type, idiomorph, which has been discovered in the Pacific Northwest. As I said, it emerges as a flag shoot in late winter to early spring, and you can see that over on the picture on the right. But sometimes the emergence can be asynchronous, and after growers go through and prune all their plants, you may still have a flag shoot that arises. These are located focally and in a very low frequency, rather rare events. At only 0.05% of plants will have a flag shoot that we detect. So I can show you a little bit more about how the fungus moves from these flag shoots in the early spring. This is some mapping data that I produced from ArcGIS. Each of these dots here represents a hop yard in the Willamette Valley. We have the Willamette River running over here on the left. And as the dots change from white to pink, those pink dots indicate that we detected disease there. And in April, these 4 dots that you see are all flag shoots. Now, in May, you have similar levels of disease. And instead of having only 4 flag shoots, we have roughly 7 here. Some of those may be from secondary spread, but chances are most of those are flag shoots. And then in June, we see a large increase in disease incidents that we're able to detect. And we also see similar levels in July. So this begs to ask, well, how does the fungus disperse and move throughout the season? And I'll show you what this looks like year by year before I get into my actual research. So in 2014, we have the percent of yards with powdery mildew on the left, on the y-axis, and then the x-axis is for each month. So as I said before, in April and May, there's relatively low levels of disease, and then there's a large increase in June, and then similarly in July. Then in 2015, we see that same pattern, a large increase in the amount of disease that we detect between May and June. Similarly in 2016, as well as 2017. Let me go back to that map real quick. And you can see that we focus kind of on the eastern extent here. And the reason why is because the western extent did not have historically as much or any powdery mildew that we were able to find. And certain yards here may end up escaping disease. And so we want to know why that is, what's the difference between these eastern sites that tend to have a lot of disease and these western sites, as well as over in St. Paul here. This is a large area for hop production, but as I said earlier, it does not have, did not historically have large disease levels there. That leads me to my objectives. My first objective is to develop a specific qPCR assay for sampling airborne inoculum of Powdery macularis. Then using this assay to correlate airborne inoculum density to regional disease incidence, quantifying those east to west differences that we've seen, and specifically focusing on the disease levels in the late season. Then the third objective would be to assess the effectiveness of late season disease control practices. So I'll go ahead and talk to you all about that first objective now, and I'll tell you what has been done and what I have done over my year here with Dave so far. So there was a previous assay that was developed in both ITS and IGS regions. Amy Peets and Walt Mahaffey developed a qPCR assay previously in ITS. There tends to be, though, a high degree of sequence variation in Powdery macularis, and that assay had a small amount of cross-reaction that occurred with Powdery clandestina, or powdery mildew of cherry. And then the IGS assay developed by Ching at WSU was not reproducible and non-specific targets amplified. So we wanted to find something that was specific, and so we looked into the beta tubulin region. We developed a primer pair and hydrolysis probe for the beta tubulin gene using sequence data from Ellingham et al. His work showed that this gene, along with a few other single copy genes that he researched, were phylogenetically informative loci for powdery mildews. Our amplification ended up being specific when compared with other powdery mildews. However, it was not sensitive enough for our applications, And I can show you what that looks like now. So here we have the relative fluorescence of an assay for hot powdery mildew in the beta tubulin region. And then these are the amount of cycles that we've gone through. So when we were using 10,000 canidia, the triplicate that I had ran would amplify well and had a relatively tight group. The 10,000 canidia was a little more inconsistent, not as efficient as 100,000. And then the triplicate with 1,000 canidia, amplification was inconsistent, and under 1,000 canidia, we weren't getting detection. So this assay proved to not be sensitive enough to detect low levels of disease in hop yards, which is what was necessary for our purposes. So we decided to move on to the LSU region. We're focusing in on the 28S large subunit rDNA, and we're developing this assay based on the MIQE guidelines. This 28S rRNA is a component found in all eukaryotic cells, and it's highly conserved. So we were hoping there wouldn't be a lot of sequence variation in hot powdery mildew, and we're focusing in on the genes encoding for that 28S rRNA, which is known as the 28S rDNA. Furthermore, we'll be using an internal control for testing inhibition following the work of Frank Martin. And the developed assay will be based on an LNA probe to help increase specificity. And I can show you what my current design looks like. So the hot powdery mildew isolates are in brackets over here on the left. And as you can see, we have one SNP over here on the right where my probe is positioned. And this was conserved between all hot powdery mildews that we tested, and then all other local powdery mildews that we gained sequence data for ended up having a different SNP. So this looked pretty promising. So we ended up testing this assay, and unfortunately there was some non-specific amplification with this design that we had. And this is a relatively common thing, as you're more likely to have non-specific amplification when you're only working with one SNP. So the hot powdery mildew is here in green, and then cherry powdery mildew, which consistently seems to be a problem organism for cross-reacting, ended up cross-reacting here. So that leads me to the limitations of this objective, and that would be, first off, the sensitivity of the assay must be able to detect 10 to 25 canidia consistently, and we only have one conserved SNP, as I had mentioned before. This is a C in other powdery mildews, and then it's a T in the hot powdery mildew. So a redesign is necessary to obtain specificity. And then another limitation here is that Podosferum macularis in the Pacific Northwest, it's a haploid genome, and it's also uninucleate. Therefore, single copy genes may not have enough DNA for low levels of detection. So most of that Ellingham data we aren't able to use for the sensitivity that we will be needing. So this leads me into my second objective, which is using this assay to correlate airborne inoculum density to regional disease levels, disease incidence, and quantify east-west differences in pathogen levels in the late season. So my experimental design, we developed a east to west transect to establish the extent of powdery mildew occurrence. And we set up spore traps in a quadrat here on the eastern side, in the center, and then in the western side. And then we have spore traps around this easternmost area to see how powdery mildew levels change at distances away from that central site. We're also doing this along with area-wide sampling that was conducted in 2019, and we're including a census of the hop cultivar Nugget. Nugget is a susceptible cultivar, so the idea there is that if powdery mildew is in the area, hopefully Nugget will have it and we can detect it that way. So here's a closer view of those quadrats. We have a spore trap sitting roughly in the center of each one of these at the top of the 18-foot trellis. And then we're also sampling by hand all the yards around and inside these quadrats. These are roughly 2 kilometers in diameter. And hopefully we'll be able to capture spores at different levels and see how those levels change throughout the season. This is the spore trap we're using here. We've done some testing to optimize the rotor rod lifespan and the extraction methods. We'll be deploying these at the beginning of May next week, coinciding with our area-wide sampling that we'll be doing. The extractions will be conducted using the nucleospin to plant extraction kit and methods from Kunjetti et al. And Klosterman et al. So we do actually have some data from the area-wide sampling that we did last year, and I'll show you what it looks like in July. So we focus in on this northern extent and then that western area that had previously not been surveyed. And we're using this to help us understand why we see so much higher disease levels in this eastern extent, and then how this changes based on longitude and in this western extent in correlation with that spore trapping that we'll be doing. So we do have some preliminary data on that as well. So this is the percent of plants with powdery mildew on the y-axis and then longitude on the x-axis. So this would be the eastern extent over here on the right side of each graph, and then the western extent of the hop growing region in the Willamette Valley on the left side of the graph. So some important things to note: we're seeing really low percent here in April and May, under 1% of plants that we sampled at each of these individual yards had powdery mildew. And then as we move into June, there are certain yards, for both June and July actually, that have 100% of the plants that we sampled had powdery mildew. And this goes to show that even though we are detecting higher disease levels on the eastern side, we are also detecting it on the western side. And we want to know when that kind of flux of powdery mildew growth, when that occurs, if it occurs at discrete time points or if the inoculum levels slowly continue throughout the year. So that leads me to talk about some of the limitations I've experienced doing this. And so we have very restricted positioning of the hop yards, which limits kind of the transect placement. As you saw, it wasn't a perfectly straight line. And then it also creates issues with cultivar variability in each of those hop yards. And then those different cultivars and those the hop yard density can potentially be confounding, as there are varying races of Potosphaera macularis that may not infect nugget. And then we can't control the landscape effects. So we're kind of at the mercy of the hop growers with what they want to plant, where they want to plant it, as the brewing market is constantly changing as the consumers want newer, more fruity beers, or they got to change out their cultivars continuously, and we can't control for that. Then the placement of trap and weather conditions may affect levels of conidia collected in traps weekly, which would then affect the analysis in the assay. So I'll do a brief little mention of kind of some of the predicted analysis we're hoping to get out of this portion of the study. And that would be doing temporal and spatial analysis of disease levels through both the 2019 and 2020 season. That would kind of look like using the relative area under the disease progress curves for each month, and then associating that with CQ values from the assay. And then the analysis will hopefully allow us to determine if those levels of inoculum in the field increase at those discrete time points or continuously, as I said before. And then we also want to know if it's increasing or decreasing, if that's correlated with weather data, whether it be warm temperature events, cold temperature events, or wind, as this is a airborne disease. And then for the assay specifically, the analysis will consist of assay efficiencies, such as specificity, sensitivity, and then the standard curve, and then also detection limits. So that leads me to talk about my third objective, but unfortunately I don't have a lot of time to talk about this. So you'll have to come back for my thesis defense in a year or so to learn more about the effectiveness of late season disease control practices. And then very briefly, just to kind of wrap it up here, so I'll be continuing to work on the development of the qPCR LSU assay, as well as conducting that nugget yard census and area-wide sampling across Oregon. And then later in the season, hoping to still be able to do the late season fungicide application in the cultivar nugget and work with that trial, and then subsequent color rating and chemistry afterwards. With that, I'd like to say thank you to my lab group. Dave, Michelle, and Nancy have done a lot of help with me in the lab. And then Brianna, Mary, Teddy, and everyone really tromping through fields and looking at hop plants for powdery mildew. Additionally, I'd like to thank my graduate committee, Nick Grunwald, Walt Mahaffey, Chris Munt, and Sean Townsend, and all the hop growers and hop research council that have supported my research. And then lastly, financial support has been provided by the USDA, Oregon Hop Commission, and Oregon State University. And I'd like to thank you all for listening. And if you have any questions, feel free to go right ahead.
All right, Cameron, thank you. We have questions for Cameron?
Yeah, Cameron. Who wants to go?
Go ahead, Walt.
I noticed in the last season you had disease on the eastern side that you hadn't really had in years past. Is that because of new plantings?
So all the data from July 17th and before that, we hadn't done a lot of sampling there, and from what I've heard from the growers, a lot of their first introductions on that side were in 2017 or 2018. They had maybe one infection, and then over the course of 2019, it kind of bloomed out there, and a lot more disease was found. So I think it's both we weren't sampling there, and then it's also was newly introduced. So it's not necessarily new plantings as much as it's the disease has just kind of slowly been creeping over to the west, and it finally made it and blew up in a big way.
Because it's when we were doing scouting for hops way back when, the eastern side had lower disease pressure unless it was a newer hop yard.
Yeah, so where we actually ended up finding disease, they were some yards that had been in for quite a long time. I don't know if maybe they put in other new yards around that area and that could have caused it, or if it made it over on some machinery. I know some of the growers that grow in St. Paul also grow in places with higher disease pressure, so they could have moved something over there. But at least the yards we found in that 2019 map, those were some older yards that had been around for a while. And I think that I know one of the growers specifically told me the first time he saw disease was 2018, and it was just a little spot. And then the next year, like 3 or 4 of his yards had disease.
So Cameron, Tom Wolpert here. Just curious, I mean, I think you're kind of touching on it with Walt's question, but can you give us some kind of perspective about how many different cultivars we're talking about here across all these hop yards? And also, you know, how long do people usually keep a stand of hops going before they replant or change out cultivars?
So it depends, obviously, from place to place. Some places are going to be a lot more dense, but one of the reasons we ended up sampling Nugget is was the most widely planted cultivar. And so here, let me see if I can't pull it up again here for you. So when we're looking at this 2019 map here, the disease we're seeing over in that western area, they have a bunch of different cultivars ranging from Nugget to Perlay to Willamette to probably 10 or 12 different cultivars. And that's a part of the reason we chose Nugget, is because we are consistently seeing disease on Nugget due to its susceptibility. To answer your second question, the amount of time that the hop yards stay in, it can really depend. You know, some of these yards will be in for 10, 15 years, because it takes 2 years about to have hops produce full yield in Oregon, so they can't just rip them out all the time. But it's also based on contracts, so they're accepting contracts right at the beginning of the year for Nugget, and if they go 2-3 years and they may not have a Nugget contract, they're gonna rip that out and put in one of the newer cultivars that might be better for some of these microbreweries, like Citra or some private, because Nugget, like Nugget and Cascade were public varieties, so anyone could grow them. So they might end up going to something that was done by a private breeding company. It's very variable between people, but there's a lot of cultivars in these highly dense areas, which is why I mentioned it as a limitation, to kind of be frank about that, because you'll see a lot of variation there.
Thanks.
Cameron, this is Joey.
Hey, Joey.
On your qPCR primer that you're designing for the large subunit.
Yes.
And you were showing that 1 point mutation that differentiated your species. Yeah, this one right here. And so these, when you're designing that probe, that primer, have you designed kind of like a sliding window approach, where you're designing up and down the length of that sequence, trying to find where maybe the most vulnerable position is to disrupt that priming?
Yeah, so I actually have ordered 2 more probes for this that I'm working on testing when I can. But yeah, that's kind of what we're going for. So I've done a redesign here where I've moved this probe, LSU probe 1, I've moved it further down, closer to the reverse primer. I've also tried moving the probe into the center there where there's some other SNPs, and then hoping that this reverse primer, if I move it on that one conserved SNP, that might help confer some of the specificity there. The real issue here is both with cherry powdery mildew and strawberry powdery mildew. The SNPs aren't really conserved between them, so where I might find specificity for cherry powdery mildew, I'm most likely not going to find it every time for strawberry. So that was the one SNP that I've kind of slid around a little bit more, but I've also done some other designs in some other places, yeah.
Okay, thanks. Any other questions for Cameron?
Yeah, I've got a question.
Go ahead.
So every time I hear a pathologist talk about spore traps, I get sidetracked into this other line of thought, which is this is a great sample for addressing one of the hardest problems in ecology, which is measuring dispersal limitations. Because you guys are getting all kinds of spores in there, not just Canidia or powdery mildew. And I thought, well, you know, you're getting lots and lots of samples, and I don't think qPCR is probably the way to go to find these, you know, a single ascospore is something that came over from the coast. But what do you think? Is there potential for reusing your DNA extractions for basically detecting rare events?
I do think that there is some potential. The issue is every different type of spore trap kind of has its own limitations. So, for example, I'm not having in-field detection right with this. So if I'm looking for a rare event, say I've set up spore traps in Australia. Australia doesn't have hot powdery mildew. So we're looking for like biosecurity and protecting their plants. I'm gonna have to sample this. I'm gonna have to go back to the lab. I'm gonna have to do the extraction. I'm gonna have to I'm gonna have to test it, and then by that time it could be in a week or so, and another generation could have popped up of powdery mildew. So I think that there needs to be some improvements with doing potentially like lamp assays in field if you're looking for some rare events. But that's there's actually a really great article that I could send you. It's about biotrapping or biosecurity in spore traps, and how it needs to be improved to detect those rare events.
Yeah, that'd be great. That sounds right on. Yeah.
I'll pull it up right now and I'll send it to you.
Thank you.
Because I haven't really researched too much about biosecurity. So even though that there is potential, it also needs some improvement for sure from what I've read.
Great.
All right. Well, let's all unmute and thank Cameron for his talk.
Great talk.
Thank you, Cameron.